All right, hi, can you hear us? Luba, can you hear us?
Yeah, I can totally hear you. Yes.
Yes, we hear you.
We can't hear you now. All right.
You can hear
We
us?
hear you, yeah.
Okay.
It's off or on? Can we hear Luba, can you say something again?
It's so nice to see you.
Yeah, here. How about now?
Yeah, it's totally fine.
Tuba, say something.
Yeah, that's what I'm saying. I'm saying something.
I know. We are
He
looking
just doesn't have any words, speechless. William, change the volume.
Too much teaching. He's like, go to the next one.
Yes, about this.
Luba,
Yeah.
can you say that?
Yes.
You gotta go to
I
the
said
settings, Mihaly.
we are looking forward to hear your
I don't
lecture.
know that I've got it. I'm just
Do you hear me?
I think they can't.
okay. And help us again. Just keep saying something. Just keep singing a song until we can hear you.
Hi, William. Can you hear me?
Yeah, but
Volume.
At least I hear you very well.
We can't hear them.
we cannot hear them.
All right.
It's hard. God, is it?
Want to say something?
Did you say something again?
Hi, William.
Thank you. All right. Hey, all right. You saved the day. All right. Thank you. All right. So now we're ready to start again. Thank you all for your patience. Thank you all for online for coming, and we have a great audience in the room today. So welcome to the Dean's Digital Cafe. This is probably our last one for the fall 2025. I'm William Letsu. I'm an assistant professor in the Biological and Chemical Sciences Department here at New York Tech. I'm excited to welcome Zakir Hossain. Zakir is a chemistry PhD student. He's in our first PhD cohort. He keeps coming to us from CUNY at Queens College. And he's now in Dr. Subha Chaudhry's lab, and he's working on pyruvate carboxylase. So Zakir has been in one of my classes this term. He's been an excellent student. I've learned a ton about pyruvate carboxylase from him. I hear he's also an excellent TA or lab director over in the city, and a lot of you are his students. So we're glad you're here today. And with that, I'll leave it to Zakir to tell us about pyruvate carboxylases and studying cancer cell metabolism.
All right, thank you, Professor Letsu.
Yeah.
Hi, everyone. Hope everyone is doing all right. Thank you for joining today. Excited to, speak about some very interesting information. So the title of today's talk, Okay. Okay, so the title of today's talk is Development of Pyruvate Carboxylase Inhibitor to Probe Cancer Cell Metabolism. So, okay. So why is this important? So as we know, cancer has a very negative effect on human life. It not just affects the person who actually has, but it also affects the people are surrounding them emotionally and financially. So it is one of the biggest factors in terms of mental health in just everyday population in America and throughout the world. So our job is to educate. By education, we can at least get people to think about early diagnosis of cancer, but also to start thinking about what is cancer. Everyone has an idea of what cancer is, but even now, in some research and books, people still say we don't know what cancer is. So education is very important for early diagnosis, and also to help us understand how cancer metabolism works. So researchers such as myself and others, are still studying cancer, right? Cancer is it's a big field. We want to be able to, not just destroy, or eliminate cancer, but also study it so that if something like this were to appear moving forward, can we use the same understanding of cancer to help diagnose other future diseases? So this is why our research is focused on understanding the metabolism, understanding how cancer proliferates and how we can control it in terms of inhibiting, but also what makes cancer cells grow. So our project is to look at pyruvate carboxylase. Pyruvate carboxylase is a mitochondrial enzyme, and it has one very important role. So PC catalyzes the reaction from pyruvate to oxaloacetate. And this oxaloacetate is very important in the TCA cycle. It is a precursor that helps with it is a oxaloacetate is a precursor that helps other synthetic pathways in TCA cycle to create precursors for other biomolecules such as amino acids, fatty acids, and glucose, which is very important because in cancer cells, right, glucose is needed a lot for it to proliferate and for it to metabolize. So we are focused on PC because PC has a very important precursor to the TCA cycle, and hopefully we can understand how that controlling oxaloacetate or inhibiting PC or even activating PC controls the metabolism of cancer cell. So PC is very important in both the catabolism process and also anapolaric process because it helps in synthesizing oxaloacetate. Now, why is PC important, right? We have to at least understand how PC is regulated or regulates cancer cell growth, right? So, one more. So PC has been seen overexpressed in these cancer cells, such as breast cancer cells, non-small cell lung cancer, colon cancer, and various other cancers. So we want to see if regulating PC will actually stop to help stop the proliferation of cancer cells, and also to see if we can slow down the growth, right? And so we did let me just move this out the way. So we did
minus the whole minus might go away.
So we did a knockdown experiment of PC in metastatic breast cancer cells, and you can see that by knocking down PC, we're able to decrease cell proliferation. And so the gene sequence we use is silenced RNA sequence to show that if we're able to control PC, we can stop or slow down the growth of cancer cells. And this is just explaining how oxaloacetate helps how pyruvate carboxylase helps synthesize oxaloacetate, which goes on and helping to feed the TCA cycle. And the TCA cycle is important because in cancer cells need a lot of energy, right? And to perform that energy, it uses the TCA cycle to create precursors to help create other biomolecules to help in replenishing its energy source. And PCA does that by synthesizing oxaloacetate, which is one of the main biomolecules for the TCA cycle. So we see that PC in itself is a very important molecule that's contributing to the metabolic process of many different biosynthetic pathways. So PC is also coincides with the Warburg effect. So what is the Warburg effect? The Warburg effect is in oncogenetic cells, it actually disobeys the regular function of the cell and starts doing gluconeogenesis, meaning that it starts synthesizing glucose for more energy. So what it's doing is actually stopping the regular metabolic process of cells and doing the opposite. So it can start creating more energy for cell growth. And PC helps in that effect. And we see that aberrant PC activities are linked to several other cancers, but also we see that it also leads to type 2 diabetes and also listeriosis. So we have a good starting point or a good enzyme to start with PC. And also, there is not much known about PC. There's other factors that we can affect in terms of controlling cancer cells, but PC is very new. There's not that many known inhibitors of PC. So it is a very new field that we're trying to figure out, or a very new enzyme that we're trying to figure out. Okay, so now that we introduced PC, let's actually talk about PC itself, right? What is PC? So PC is a mitochondrial enzyme. It has 4 identical subunits. Weight is 130 kilodaltons, and it's an ATP-dependent enzyme. So what is this? And it's also found in the mitochondrial membrane. So it is important to understand this because this being in the mitochondria also has a disadvantage, right? So we have to go to 2 layers we have to go through an extra layer of membrane to at least get to the mitochondrial membrane. So we have to be able to bypass the normal cell membrane layer. And then after it bypasses that, we have to get to the mitochondrial membrane. So we're looking at 2 different, 1 extra hurdle that we must pass along with a bunch of other hurdles to understand how PC works. And so the reaction you see here is the main reaction that's actually being catalyzed by PC. We take pyruvate, bicarbonate with ATP, and it catalyzes, synthesizes oxaloacetate. So what is the structure of PC? So this is the monomeric form of PC. PC is made out of 3 main subunits. The biotin carboxylase domain, the carrier protein domain, and the carboxylate transferase domain. And then we have another minor domain, the allosteric domain. This domain is not a part of the main mechanism of reaction for PC. And so what happens in the BC domain is the reaction where biotin, where the carboxylate group from bicarbonate is added to biotin with a magnesium ATP dependent cofactor. Then once the biotin is added, ATP is hydrolyzed to ADP, and then this reaction occurs in the BC domain. And then once this reaction occurs, the carrier domain takes biotin carboxylase and then shifts this to the carboxylase transferase domain. And in this domain, we get the addition of the carboxyl group to pyruvate to get oxaloacetate. So the monomer form of PC is not the active form. The active form of PC is a tetramer. It's 4 identical subunits that combine to actually facilitate this reaction. So keep this in mind moving forward, as this is important aspect to know for our research that we are trying to explore. Okay, so we know about PC. So how do we study it? So our first generation inhibitors that we synthesized were the alpha carboxylic acids. So these, when we are looking at inhibitors, we're looking at 3 important factors: potency, selectivity, and permeability. These three factors must coincide for us to look at or understand the how PC works. So our first generation inhibitors, or instead of saying inhibitors, I would like to say modulators, because we're not, our goal is not to actually focus on making inhibitors now. We're actually trying to find modulators to see how PC is affected by the different compounds we are making. So our first modulator acts as an inhibitor. It is the alpha enol carboxylic acid, and it has very good potency, where the IC50 is less than 10 micromolar. But there are issues with this. One is the selectivity, right? It is not selective towards just PC. It does affect some other metabolic enzymes and other biotin cofactor enzymes, and also low permeability. The log PAP of this is -8. Log PAP is just a criteria that's used to see the permeability of molecules. And so this is a negatives, sorry. A value of -5 or higher means that it's low permeability. And we can see that even in the structure, as this group here cannot actually go through the cell membrane, because as we know, the cell membrane has a hydrophilic and a hydrophobic region, and the carboxylic acid and the enol form of it is not good to go through the cell membrane. So we have to now think of another way, right? We know we can modulate it by this molecule, but we have to think of another way to affect it or to see the inhibition of it. Because PC itself, if we in our future goal is to make a good drug candidate, we don't want to stop PC's activation, right? Because PC is still needed for regular cells to work. We have to be able to stop it, right? To stop the cell growth, and then also be able to reactivate it to act normally. So we can't really shut off PC forever. We need it for regular enzymatic functions. So we need a new class of molecules that have good permeability. And so these molecules act as other ATP modulators, PC modulators. Analogs are pyruvate. They require a big dose of the drug. So these drugs, once you have big doses of it, they can start affecting other enzymes. And so these are not very good candidates to use for future work. And also, as I said, it's non-selective. And then the higher concentrations start affecting other cellular processes.
But Zakir, does it matter whether it affects other cellular processes? I mean, ultimately, you want to kill the tumor cells, right?
Mm-hmm.
So what difference does it make if it attacks other cellular processes?
Well, so if it starts, then it's not selective, right? We lose the selectivity of it. If it starts affecting other cells, then how do we know that if it's really affecting PC, right? We want our compound to be selective also. We don't want it to, we don't want to have off targets, right? If it has off targets, who knows how the drug is metabolized in that process. So we want it to be selective. You don't want it to affect more than what you're trying to affect. Because if it starts affecting other things, then you have to worry about that. And which, too many variables is not good.
And also worry about affecting other cells of the body, right?
Correct.
Right.
Yes. So that was our first gen. So our first gen inhibitors was, the best one was this alpha carboxylic acid. So we have to think of something else that helps with the permeability aspect. So how do we do that, right? So first, going back to our main goal was to increase selectivity, make it only selective to pyruvate carboxylase, increase potency. Potency in terms of not just inhibition, but to see if it also greatly increases the activity of pyruvate. When we mean potency, we don't think about just inhibition. We want to understand what is also the activators, right? Is it a very strong activator PC, a weak activator PC, or is it a strong inhibitor and weak inhibitor? So we're looking at both spectrums, and then also permeability. As I said before, it has to go through both. It has to go through both bilayers for it to get to its targeted enzyme. So how do we do that? So we do that using in silico screening. So what we did was with our collaborators at Atomwise, they took about 2.5 million drug candidates, and using that we did virtual screening to see which one would fit the CT domain. So in terms of homology, the BC domain is homologous around all by all PCs and all species. It is the CT domain that has some differences, and that is where we're targeting the reaction, because the main reaction that's occurring in PC is at the carboxylase transferase domain. So we are our main goal is to study how the inhibitors affect the CT domain activity. And so what we did was we took about in virtual screening, we took about 2.5 molecules and did a screening on which one would bind to the CT domain. Once that was done, we went to roughly, I think, 30,000. And then from those 30,000 molecules, we did another criteria of screening using the Lipinski 5 rule and the Payne's rules to remove those. And from there, then we got about roughly, I think, I believe close to 100 molecules. And those 100 molecules we bought. And our collaborators at Dr. Martin's lab in Marquette took those molecules and did a assay. The assay they used was a fast violet blue assay to test those molecules. And after testing, they found one molecule that had double-digit micromolar activity, which is our lead compound, the IZT. And so they also did this too with another actually. The FVV assay is only special to our group, but we also know another assay, the malodehydrogenase assay, that is also used to study PC activity. And to verify that this drug is not special to our assay, we also tested with the MDH assay, and they both say they have the same data. So this is the 2,4,5 imidazolidine trione is a good candidate. The handful, so that's why we call it ICT.
So were all these chemicals that you screened pre-existing drugs, or were they any like novel compounds? Because you mentioned you bought all of them.
So these were not novel compounds. These compounds are already known or on the market, or already drug candidate. Not all of them, but some of them are drug candidate worthy. After the first 2.5 million are screened, then when we get to 30,000, then at that position we actually do the drug candidacy experiment, where there's a certain rules and criteria that a drug should have to be considered a drug candidate. And so that's where we crossed out a lot of them, and we got to our 95, about roughly 100 compounds. And that from those screening, then we actually bought those compounds, and then they did the in vitro screening. So they tested all these compounds in different micromolar activities, and saw that this IZT had double-digit micromolar activity, so low micromolar activity, as seen in our first in our first generation inhibitors. Okay, and so this is the candidate that we found after the in vitro screening, and it had an IC50 of 40 micromolar. And so just to go over some of its characteristics, so it's disubstituted imidazolidine trione. So the trione is referring to the 3 carbonyl groups, which is our main scaffold. And then we have 2 R groups. The first R group is this I don't see it. I had the name of it in my head and I was just gone. It's not tetrahydrofuran, but it's the thio version of the compound, and then the methyl group. And so, in short, we just call it substituted IZTs. And so, now since we know that, now we actually have to make various analogs of this compound, right? So we want to increase, like I said, we want to increase selectivity we want to see the potency and also see the permeability. So once we have that, we make various class of compounds.
Oh, so you know the reason I'm going over there to Mexico? Wanna know why? Why? Because my fucking roommate, we go to do groceries together, right? Yeah. And I buy like this drizzle thing for my.
There we go. Just talking. Guess his roommate is not a good help.
That was a good introduction. Okay, so after we developed all these compounds, then we found our new lead compound, which was I think don't have the name of the compound, but we have the main important features of our compound, which is the ethanoic group here and the phenyl group here. So knowing these information now, we can start making an SAR, which is a structure activity relationship, to see how changing these functional groups will affect those three parameters that we want to see in PC inhibition. And so the IC50 is about 3 micromoles, which is a very good starting point. So this is really not the endpoint. This is where we start. Now to see this, but there's also some important things we have to understand about this, because pharmacologically, esters are not the best compounds to use moving forward, because they metabolize differently in the body. So we have to also fix this group, right? And to see, we also have to see the permeability. Just because we saw this in screening, we have to check the permeability of it. We know the potency is there, right? We also later on also talk about the selectivity. It's also selective and not affecting other metallic enzymes or other biotin carboxylases. So we know it's selective, but is it permeable, right? And so if you look over here, the permeable data, well, we said over here, it doesn't inhibit other metallic enzymes or biotin-dependent enzyme, but the permeability is not bad. It's -7. Close to -5 is ideal, but -7 is not the worst. So we're at a good position now to make different analogs of our target molecule. And so just comparing our results to our first generation, we know the potency is very similar. If we go back to the potency of our first gen.
Zakir, before you go to the potency, just for the permeability, how did you check the permeability to determine that it was that -7? Is it in silico also, or
So
actually physically did an experiment?
Yeah.
Where...
Well, I didn't do it.
Yeah, right.
Our collaborators, yes.
Yeah.
Our collaborators did do a permeability assay to see that it was had that what do you call it? Had that number.
Wow.
I don't know exactly how they did it, but
Okay.
yeah, they did do that. Oh, sorry, I was trying to go back to this. Yeah, so you can see that our first gen had a very good potency of -3 micromolar, and our new gen has not negative, sorry, 3 micromolar, but and our new gen also has the same potency. So we are equal in potency, but we have better. For this, the selectivity is better, right? Because the other one we saw that the selectivity was not only selective to PC, but this compound is selective to PC and not other metallo having enzymes and biotin dependent enzymes. And also the permeability is better than the compound. And you can see that this compound itself structurally is a very non-polar looking molecule. So it can pass through the lipid bilayer more easily compared to groups that have more polar groups. And so we were able to at least start checking off the boxes with our new class of inhibitors, right? So the potency was the same to our first gen. We increase the selectivity and we increase the permeability. So now we're at least going in the right direction in terms of what we would like to see. And so we don't stop this. So now we actually have to get more information from it, right? So we have to at least now start creating the SAR. So we want to see what amino acids are close to the active site, or if it's not even the active site, is another site. We first have to understand that. Is it at the active site or not at the active site? And to that, we have to figure out where the compound is binding. All right. So if you look at the Lineweaver-Burke plot, right, which is also a double reciprocal plot, we can see that compared to pyruvate, our molecule is a mixed inhibitor, but to ATP, it is a non-competitive inhibitor, suggesting that this is really not at the active site of where pyruvate, where the carboxylate is adding to pyruvate. And so we want to understand where is it now binding to get better information about the structure of PC. Now, we've been talking about PC for so long. I'm guessing you guys never thought about what are we using PC? Where is this PC coming from, right? So much of this PC are coming from bacterial PC, especially Cephalosporin aureus PC. And our in vitro studies were actually in Rhizobium etli, right? So we use those different types of PC for studying because they're easier. Well, I don't want to say easier because I've never done it, but in terms of studying it, it is easier to deal with those bacterial PCs for our studies. But we also want to study it in human because ultimately that is our goal, to see how PC is how PC controls human cell cancer metabolism. All right, so where do we think it's binding, right? So we know it's non-competitive, and we know in terms of pyruvate, but it's also, it's a mixed inhibition towards pyruvate and non-competitive towards ATP. But so where is it binding? So our hypothesis is that it's binding at an exo binding site. So remember how we said that PC is a tetramer, right? So all 4 subunits must bind together for it to work. Our hypothesis, this is not actually, we haven't, made it, official yet, but we're still studying. But our hypothesis is that where our compound is binding is actually stopping the formation of the tetramer, right? So it's binding in a position where the tetramer is not forming. So that's our ongoing hypothesis, and we're still trying to figure that out. Yes.
So Zakir, you started by saying that you did the in silico screening to find out basically how many targets, molecules you can identify that target PC. So why couldn't you use the same approach, a computational approach, to find out exactly how your drug is binding to PC, right?
So that is a good question. So if I were, well, I wouldn't even want to answer that because I wouldn't know where to start. But I guess so the human PC is different than what do you call it? To bacterial PC, right? So those in silico were done in bacterial PC. So we are not really working with bacterial PC. We're trying to work with human.
No, I understand, but the structure of human PC is known because the sequence is known, so the structure is there. So why couldn't you take that structure and take the structure of your drug, put it through a computational program, and find out exactly, right? Because it's based upon the energy and the bonds and everything, right? So why couldn't you find that out computationally?
We could probably find that out computationally, but I don't think we have done that yet, which is a very good reason to move forward. We should do it, but I that's a good question. I would have to ask Suba about why we didn't do that, but I believe that
isn't this sort of what we're seeing right here? This looks like a
this is a predictor. So we are predicting that this is at not at the binding site of the CT domain. So the molecules that we did, right? We actually screened it for the CT domain, but we saw that the inhibition actually did not go up or go down, right? It stayed stagnant. So that stagnant effect is saying that it might not be actually at the CT domain. It might actually be at an exo binding site, which is where hypothesizing that this is where it's binding, that it's binding in a position where it's stopping the tetramer.
So I think Suba raised his hand.
Yeah.
Yeah. So what I wanted to say about this. So now we have been actually not me or my group, but our collaborators, they have been trying to get a co-crystal structure of the bacterial PC, which is Staphylococcus aureus PC, which we are actually using as a screening tool with some of our synthesized molecules, particularly the IZD molecules. But they were not very successful. However, they got some very, very thin layered crystals, and the resolution wasn't really great. So they are not publishable. But there is a strong indication, or you can say that there are some evidences which we couldn't really publish yet, saying that it's binding in a position which is not exactly the CT domain, but in between the CT domain and that allosteric domain. And by binding there, it's actually disrupting the tetrameric structure formation, which is the actual active structure for the catalysis. So that based on that, we did this hypothesis. So there are some evidences, but still, I think we need a good or high resolution crystal structure to prove that, right? So, but yes, again, the human pyruvate carboxylase, we didn't really do many experiments, and there are reasons that why we are not doing human pyruvate carboxylase, because when we try to do the expression of human pyruvate carboxylase on the insect cells, that isolation wasn't easy because it was not soluble. So we did some chimeric, and then the other is the pig liver PC, which is like 85% homologous. So these are the other two PCs. So we have a 3 PCs now, a panel of PCs we are assaying our compounds against. So
Yeah.
Thanks, Suba.
Thank you.
yeah.
I don't know how to raise my hand in here, really, but I just wanted to help out a little to Zakir about this atom-wise thing, because I know how that worked. Atom-wise, like a few years ago, asked like more than 200 labs if you have any structure that you want to work with. And because the idea for Atom-wise company was they had this AI machine learning ducking tool for predicting the compounds, and they want to test out how well their algorithm is. So they send like a open request to whole US, and some labs say this is our target and this is what we would like you to do research. And most probably, I'm predicting in here, the binding prediction that Zakir is showing is what Atom-wise is predicted for the lab group. And to be able to do that, Atom-wise was asking the labs, tell us where you want us to dock it. So the labs actually had to choose the domain, tell exactly the pocket, and then they would just screen millions of molecules in it. So
that's exactly happened. That's exactly happened. So our program was with Atom-wise, but we only gave them CT domains. So this is not exactly they predicted. It's our prediction. So since the CT domain was a catalytic domain, so we gave them that domain, and they screened 3 million. 3 million. The number is huge. 3 million compounds. Okay, from enamine database.
Yeah, exactly. And my question in here is that so the ducking, if it has not been done in other pockets, they basically, like Shubha says, they would not predict that binding affinity towards the other pockets, which is still possible. So
of course, that's right.
That's why like the mutation studies might be helpful in this case if you cannot get like the crystal structure. So that's one way. One way is crystal structure. We for sure predict that it is the case, but like I guess mutation studies can be done in this case if the stability is not good enough for crystallization.
What studies? Say it again, please.
Mutation studies.
Sorry, I can't.
Mutation.
Rotation.
Yeah, amino acids, because then you have to mutate you first dock in all different pockets that you have. Basically, you have 4 domains, and then you dock all those 4 sites, the most probable sites, and then you look at the predicted interactions, and one by one you just mutate each residue, and then you have a better clean idea of, okay, I changed the tyrosine to alanine in here. This is usually what they do. And then they look at the inhibition data. If it is affecting, then you just have a good say that, okay, this is the pocket that I am binding. But my question is not going to be about it. It was just for the help to Zakir. And my question would be, is the problem with the permeability and the ester functional groups? Because the compounds that has been docked in here is probably the ester that I'm seeing. Do we really know that the ester has not been hydrolyzed, and actually you're seeing the effect through a prodrug effect or not?
So this, so the hydrolysis of the ester happens in acidic conditions. So this what did not go is through its regular metabolic process for the ester to be hydrolyzed. We are for this molecule, we're just testing the ester if the ester was not hydrolyzed yet. But we know that the esters can't be the best moving forward best drug candidate because it has the potential of being hydrolyzed in our body. So this is.
So you had a direct protein inhibition assay in here?
Yes.
Okay.
Yeah, I just wanted to add this thing wherein that at this point, I know that you asked that question to Zakir, but for clarification, that at this point we are looking at the potency, not the pharmacokinetic data, right? So once we get the optimized potency and we know the kind of interaction, then we will look at the functional groups and probably do a lot of bioisoster studies to get the best group which will be pharmacokinetically stable.
Yeah.
So we are not studying pharmacokinetics at this point. So we are not even thinking of the hydrolysis of ester or anything. We are just looking at the interaction between the target and our molecule from the pure perspective of enzyme assay.
Yeah, that actually leads to my second question, because when Zakir showed us the core imidazolidine structure, and then showed that there is basically 2 ways that can be differently substituted, R1 and R2 groups. My question is, like, in the beginning to generate the structure activity relationship, how did you come up with the R1s and R2s? How did you select it and do the synthesis?
Yeah, Zakir, go ahead, answer that question. You should be able to answer that.
Oh, so usually, there's usually if we go to our lead compound, right? Our lead compound, we took some of the aspects of our lead compound and used that to synthesize the molecule, right? So sorry, that's not
No, this is not the lead compound, Zakir.
Go back. Sorry, right here, this lead compound, right? So
no, that's not lead compound either. So go to that previous slide.
Oh, sorry, yeah.
Yes, this is the lead compound.
Yeah. So this was the compound we found through the pseudo in vitro screening, and so you can see that we have a what do you call it? We have a nonpolar group with we have a carbonyl group here, and then we have a group that's very nonpolar. So just taking this information, we made various other analogs. Now there is no way of saying, oh, let's just do this. What we're trying to do is we're just taking that information of a group that has a carbonyl functionality and a group that is very nonpolar, and adding that to and then also there's other key ways to look about how you want to increase interaction, right? So you want to have pi-pi interaction stacking, or hydrophobic interactions and hydrophilic interactions. So we want to test all those interactions. And so when they synthesize these, we synthesize these compounds, we took that into aspects. So we know esters, and then we also had non-polar groups. You can see there's also derivatives of thiophenes in here. So we took all those and synthesized these molecules, and the best one came out to be this one. But it was no really, it wasn't guessing, but it wasn't also like we're going to try this. We have to try a bunch of molecules to even get to this. So we had, I believe, well over 100 molecules that were synthesized and tested before we got to this lead compound. I mean, not the lead compound, this our more potent compound.
Yeah, but like you had in the beginning, just the metal group, right? In your R1, how did you decide going with an ester functional group? Because you
right. So yeah, I think she has a very legitimate question, Zakir. So yeah, since Zakir actually joined recently, so probably he doesn't know the whole story. So what happened, after we screened all those compounds, and then we got hundreds compounds after eliminating all those false positives and the structures which are not drug-like. So out of 100 compounds, the enzyme screening actually gave the lead compound, which Zakir showed, which around 40 micromolar activity against the pyruvate carboxylase, the streptococcus aureus, pyruvate carboxylase. Now we got a compound where we have that imidazolidine trione ring system in the middle, and then two substituents. So basically, it's 1,3. We call it 1,3 disubstituted imidazolidine trione groups, where you have an R1 alkyl and R2 alkyl. So now,
Zakir, get through a little. I think we're getting a little too technical, but these are good questions.
okay.
That are really helping him. I think but I think we want to let Zakir finish his talk.
Oh, okay. But can I just say one? Maybe Berin, I think your question is very, very legit questions. But I think when Zakir will get to the synthesis, then you will see that there is a two-step synthetic process which gave us over 100 compounds, and with different types of functional groups as an R1 and R2, and based on the availability of the isocyanate and the amines, it showed that the compounds which are showing like a single micromolar potency are the ones where you have one aromatic group on one side and one ester group on the other side, right? So these are the compounds in this slide. They are the good compounds. Now, there are a lot of bad compounds which got over 100 micromolar or 1,000 micromolar. So we didn't even consider those, but they had all either, aromatic groups on both sides or aliphatic groups on the both sides. But
yeah, so there was a preliminary work behind.
Right, that was a preliminary SAR, the structure activity relation. After out of 100 compounds, we arrived at this. Okay.
Okay, thank you.
Thank you.
Zakir, go ahead.
Thank you. Okay. All right. I guess we'll just So I think I talked about this. Right. We were sorry. Okay, we were here. So binding interaction. So our prediction is binding at a different site than we were hoping it would. Okay. So as I said, this was starting with PC. Our ultimate goal is to work with human PC, and so human PC, right? Oh, sorry. Why is not both coming at the same time? Okay.
One and the other.
Okay, it's fine. I can go back to that. So substituted, we know that it's selective for S-aureus PC, and then we also did the knockdown PC using the MDA MB231 breast cancer cells, and we see that it does decrease the proliferation of the cell. Right? It does, but do not inhibit MDA2 cell viral. Oh, sorry, cell viral mean that it does. So it's knocking down PC activation, but it's not killing the cell. Sorry. Right. So now we have to actually start working with human PC, right? So as Suba alluded to, we are working with pig liver PC because it is very close to human PC with a 97% identical. And I was always curious about why we're working with pig liver PC, because it is more easily able to obtain and also able to process to get the PC from pig liver. So we chose to use pig liver PC as it is closest to human PC. And this is our Dr. Nick Schneider. He was the one who was you can see actually getting the PC, the liver. And so, but as Suba was leading to, human PC is not the best PC to work with because it has solubility problems, right? So what we did, we made a chimeric PC. So we kept the VC domain the same from because it is not the target of our study, but the target of our study is a CT domain. So we kept the VC domain from other species, and then we used human the human CT domain and BCCB domain. And then this domain is we're not really focused with, but this was also from human, and we use this to test our molecules. So it's called chimeric because it's portions of different species that we're testing. Right? So we can see that the chimeric human PC is significantly less potent to human PC. So there's still more work to be done, right? We need to make sure that it actually affects human PC because we know it works with bacterial PC, which is our initial, but we have to now create other analogs of our compound of IZTs to make sure that it's also affecting human PC or at least our chimeric PC. Right. And so we can see that it does have some effect, well, not the best effect to even pig liver PC. So we're still focusing on trying to make other different analogs to see if we can get this inhibition process to work on other various PCs. And there is a reason why this isn't working, but don't want to get into it now, but we do know why that this inhibition is less potent in these PCs compared to bacterial PC. All right. And so future work will be on trying to figure out where it is docking, but also once we know that, well, before even knowing that, we have to see to get the activity even more potent for us to be able to maybe get a crystal structure or even know the binding of our molecules to PC. And so this is probably the most fun part of this whole talk, the synthesis, okay? The general scheme that we use to make IZTs are the top ones. So we take an isocyanate, reacted with a primary amine. This will be our first R group, right? And then this R group will give us the urea. And then, the first R group is actually, attached to isocyanate. And then the second R group comes from the amine. So this can be a primary amine, a secondary amine. And then once we have that, we react it to give us a urea. And once we generate the urea, we add excess oxaloacetate. That's not oxaloacetate. Oxalochloride to make the triol. And so this is the general scheme. This scheme, it takes about 2 days. If sometimes this reaction is actually pretty fast, but to make sure that all the reaction occurs, we usually leave it overnight. But this reaction is actually, if we just kept to take the TLC, it reacts pretty quickly, and then this reaction takes overnight. So we can generate a lot of different molecules very quickly and abundant of them. This one here is still being tested out to see if we can make in an easier way, right? Because this we don't purify until the last step, but this trying to see if we can make it in an easier way. And so how do we act? So this was just a general scheme of how we make that compound of the ester and the phenol, right? So we took the salt form of the amine reacted with phenol isocyanate. Once we have the urea, do cyclization to make our compound, and we can change these R1, R2 from our starting materials. And so, but we want to do other things, right? None of these we won't have all all the different R groups that we want from just the starting material, right? So we have the other synthetic ways of synthesizing different R1 and R2 groups. And so this is one of the other paths, which is just a peptide coupling reaction.
Zakir, sorry, what is the yield of this reaction?
So the yield for a compound, so the purification of these was done towards the end of it, and for our purposes now, we're not really looking for yield. We want to really test as much compounds as possible to see the best compound. And once we get the best compound, then we can see what the yield is. So right now, we're not really keeping the yield for a lot of these compounds, but I can tell you, I don't know if it's this compound, but I can tell you for the amide coupling reactions, usually the yield is close to 50%, but since this is a multi-step, it might be a little bit less. But right now, we're not really keeping track of yield.
Zakir, this is a very good question, and I think you take a note of this, even though we are not really looking for a good yield, but I think that's a very legitimate question. So that is a learning experience for you as a graduate student. So I would ask you to take it very positively. Okay?
No, of course.
That's a very legit question.
Yeah. I mean,
so every equation you should come up with an yield, even though it's low.
Okay. And then so this is some new methods we're working on. So the carboxylic form of some of the compounds are the best to start with. So we start with the esters, and when we start with the esters, we can hydrolyze the esters to the carboxylic acid and do other various amide coupling reactions with it.
Yeah, this might also answer some of the Berin's questions. Berin, so this is also one of the reasons that we are trying to modify the ester into amides, even though the amidase enzymes could also hydrolyze amides, but that might be a better pharmacokinetic group compared to the ester.
Yeah.
Okay, and then just variations of other reactions. And so conclusions, we know some basic information of how IZTs, the potency, permeability, and selectivity of IZTs, but that's just the beginning. So we're working on making different derivatives of these just to get a better picture, because we ultimately do want to get a crystal structure of human PC with our molecules to see where we can where we can make adjustments to increase the potency of it. And so that's it. I do want to acknowledge the PhD program in NYIT. Thank you to the Dean Digital Cafe, my advisor, Subhash Chaudhry, Dr. Martin, and Nick Nick is no longer there, but Martin's lab that did all the biological work at Marquette. So thank you to them. Nick did a lot of the initial biological studies, and Dr. Donaldson, who where Suba was in before, worked on synthesizing these molecules, and then Neil Hankerson's from Adamwise.
We're at 5, so we have to go under. If we have any questions, I'm sure Zakir would be happy to answer. I do have one.
Yeah.
So it seems really important that the pyruvate carboxylase works as a tetramer, right? Does it work? So I'm wondering, does it have no activity as a monomer? And also, like, do you think this has some connection with why your compound might be fitting like in the interstices between the monomers?
Yeah, so this has the PC itself, its active form is the tetramer. So the monomer itself in the body won't do the reaction. It has to be the tetramer form for it to be the active form of it.
Like even in solution, it has zero activity by itself.
That is a good question. I don't know the answer to this one.
Because then it really goes well with your hypothesis that it's fitting between the molecules, right?
So that so that hypothesis I think was, we don't know. We think that's what's happening because we see that the inhibition is not getting better or weaker, right? It's just stagnant. So we're thinking that that might be the reason why it's not binding where we think it's binding, and it's binding somewhere else.
Can you repeat that question? Sorry, William. Can you repeat that question? I was thinking something else. Can you repeat that question?
Yeah, I was just wondering, do we know if there's zero activity for PC in the monomeric state, or is it just?
Yeah. So PC does not, for any catalyst to be active, it requires a complex, formation, right? So it needs cofactor and it needs to be in the right form. So yes, I don't think that in a monomeric form has any activity. So it needs to be in tetrameric form. Now, why we think that it's binding to an exo domain where the tetramer formation is hampered? Because in a series of molecules having one aromatic group on one side and ester on the other side is giving you a plateau of the potency, meaning that they are having like similar potency. So they are not like either binding strongly or weakly. They are giving the similar kind of potency, saying that it's only hampering the tetramer formation. Does it make sense?
Yeah, thank you.
Yeah, so because the potent for if you have a higher affinity of a particular analog, then that would probably give you higher potency. But it's not showing like that. It's just breaking down, or it's kind of blocking the tetramerization, so the catalytic activity is hampered, and that's how it's working, and that's why we are seeing that plateau. So a lot of these analogs are having like say 3 to 5 micromolar activities. That tells us that it's just disrupting the tetramer formation, and that's how it's working.
Thank you. Okay, thank you.
Can I just, before you close it, if I have time, just making 2 points, just 2 points. I think Zakir has said he has done a good job. The other reason that the chimeric human PC and the pig liver PC, they did not really show the similar kind of potency what the Staphylococcus aureus PC showed. The reason being, unlike the bacterial Staphylococcus aureus PC, the chimeric human PC or pig liver PC needs an activator to get to the right conformation, and that is acetyl-CoA. So whenever we do the experiment, we have to really do it in presence of acetyl-CoA, and acetyl-CoA is an activator. So unless you have an acetyl-CoA, this is not gonna go for the catalytic activity, and if it doesn't, then there will be no point in assaying those things. So when you test any of those analogs, you have to really do it in presence of a activator, and because you are using the activator, your potency will be a corrected potency, which is actually much less than what you see with the bacterial PC.
Thank you.
All right. Well, thanks, Suba. Thank you all for listening. We've had a great talk from Zakir. This is probably concludes our Zines Digital Cafe for the fall. We have some speakers lined up for the spring. But if you're interested in giving a talk, email me. Or if you know someone who wants to give a talk or should give a talk, also email me. And thank you very much. Have a great rest of your day.
Thank you.
All right.
Thank you.
Zakir.